Hello everyone. In this lecture today, I'm going to talk to you about Sirius Red Staining in detail. I'm going to talk about what is the principle of Sirius Red Staining, what is the protocol of Sirius Red Staining, and how to do the interpretation of Sirius Red Staining data. So Sirius Red Staining is also known as Sirius Red Staining. This is used to stain collagen network. Sirius Red Staining is used to stain collagen network. And what is collagen? Collagen is one of the foremost components of the tissue's extracellular matrix, which provides strength, elasticity, and architecture to the tissue. Okay, so Sirius Red Staining is used to stain collagen network. This is the important point. And so in bright field microscopy, so when we perform Sirius Red Staining and analyze these Sirius Red Staining sections under microscope, then we will see a collagen in red on a pale yellow background. As you can see that this red coloration, these all the red on this pale yellow background, okay? Pale yellow background, this is a Sirius Red staining, okay? So staining of the collagen in the tissue section. So this red thing here indicates the collagen content stained by the Sirius Red staining. So then what is the Sirius Red Staining protocol? The protocol provided here is the protocol for paraffin embedded formalin fixed tissue sections. So 20 minutes in xylene followed by 20 minutes in 100% ethanol, then wash 5 minutes in distilled water, and then place slides in Sirius Red solution for 1 hour at room temperature. Afterwards, rinse slides quickly with distilled water. 20 seconds in 70% ethanol and 20 seconds in 100% ethanol, 20 seconds in xylene, and finally mount the cover slip on slides using Entelan. Okay, so this is the protocol for Sirius Red Staining. So then final point is how to do the Sirius Red Staining data interpretation. You can see here we have two pictures, one representing control sample and the another picture representing treatment sample. As you can see that in the control sample we have more red coloration, more red network. So okay, this is indicating more collagen content, whereas in treatment sample we have less red coloration indicating less collagen content. So this means that control sample has more collagen content than treatment sample, and hence the control sample has less fibrosis, okay? Less fibrosis than the treatment sample. So higher collagen content is an indication of fibrosis in liver sections. Therefore, control sample has more collagen content and hence more fibrosis, whereas treatment sample has less collagen content and hence less fibrosis. And how we know the collagen content is more? Because of highly stained Sirius Red staining area in control sample than in the treatment sample. Thank you very much.